Good afternoon, everybody, and welcome back. I'm really happy to see that the numbers look similar to what we had in the morning. So most of you have returned back. Nobody has disappeared. Now in this session, the afternoon session is going to be mainly on basic principles of electrochemistry. I don't promise to teach you the whole of electrochemistry within an hour. The idea is to introduce you to the basic principles of electrochemistry which are relevant to biosensing technologies, because we are mainly looking at electrochemical biosensing, and I think electrochemical biosensing is what forms the basis of my subdermal wearable devices. So we're going to be looking at the basic principles of electrochemistry that are relevant to the biosensing technologies. Okay, in the morning we did have a look at the introduction to the epidermal wearables and the subdermal wearable devices. We did an outline session in the morning. Now we're going to be covering this one, which is basically on basic principles of electrochemistry. The learning objectives that I have for this particular lecture is mainly for you to understand the core electrochemical concepts. We're going to be looking at redox reactions. Redox, as you all might have already, it's your basic GCSE level 10th standard, 12th standard chemistry, where you've studied about reduction and oxidation in one word, which is called as redox processes, redox reactions. You're going to look at the electrodes, the different kinds of electrodes, the different electrode setups that you have in an electrochemical cell. And you're going to look at the potentials, and then we're going to look at how your electrochemistry is going to underpin the biosensing mechanism, and we're going to be looking at reviewing some of the techniques that are normally employed as part of the electrochemistry. This morning somebody asked me what is chronoamperometry. Well, I said again, measuring current over time is chronoamperometry. So we're going to be looking at some of those techniques here. We're going to be looking at potentiometry, amperometry, voltammetry, and we're also going to be looking at impedance measurements. And we're going to explore the applications in wearable biosensor devices. How can we normally apply these principles and get our wearable devices? I do appreciate not everybody in this audience has got enough knowledge on electrochemistry. Not everybody studied electrochemistry. Not everybody is a chemist here, and not everybody knows about biosensors. So please be patient. I think slowly as the course progresses, you will learn more and more about the basic principles of biosensing technologies, especially the electrochemical principles that underpin the biosensing technologies. All right, suggested reading. I mean, this book, if anybody has managed to read this already, I mean, hats off to that person. This is the Bible of electrochemistry. It's a book written by Bard and Faulkner. There's also other books written by Joe Wang as well. I would strongly recommend that. I've got a DOI for you here. I think you can get the online version of this particular book. It's good for looking into the principles you're working on and why certain techniques are good and certain techniques are not very useful. So it depends on what you're measuring and how you're measuring. That decides on what electrochemical techniques you're going to be employing for your for the biosensing application. All right, now we take a step back, and this is going to delving down very much into fundamentals. When I was sitting here, I heard a few people mention that they're not from the field of biosensors. So this is here a few slides to help you understand exactly where biosensors sit in the real world. A chemical sensor, people, and everybody understand what chemical sensors are? Do we have example of chemical sensor in this room? We should have one, right? We should have an oxygen depletion sensor somewhere, which should basically tell us or send an alarm when there's oxygen levels drop down. Or we should have a carbon monoxide sensor. Normally households are meant to have carbon monoxide sensors so that if carbon monoxide leaks, you have an alarm, basically evacuate, right? That's how a chemical sensor. What a chemical sensor does is, or what a chemical sensing device is, it's a device that transforms chemical information, mostly in the form of concentration, of a specific sample, and it takes it and gives you an analytical useful signal, right? When you talk about the sensors that you have in the room, you don't get to read any numbers there, but you get an alarm there, and that alarm is good enough signal for you to evacuate. Sometimes when you look at the example of blood glucometers, you have a reading there, right? So you basically have a device which converts or transforms chemical information into something that's useful, that can be analytical. For example, a reading of blood glucose, that's telling you about what the blood glucose levels are. And normally these devices will have two components which are connected in series. The first one is what we call as a molecular recognition element. It could be a chemical, and that's the heart of your sensing device, because it's that part which gives your device specificity towards the target analyte that you're looking into. And that part is also called as a receptor. And the second part that you have is called as a physicochemical transducer, the one that's involved in basically once you've identified the target molecule, and then to give you a signal that corresponds to the concentration of that target biomolecule. That's what a physicochemical transducer does. It transduces, or it does a transduction, it does signal transduction of the chemical concentration into something which could be an electrochemical transduction or into an optical transduction. That's what normally happens. In a biosensor, your molecular recognition element is something which is very biochemical-ish kind of thing, which is involved in a biochemical mechanism or involved in a chemical mechanism. That's the only difference between a chemical sensor and a biosensor. Biosensors are normally where you have, instead of a receptor, which is your molecular recognition element, which is the heart of your device, which helps you in giving you information specific to your target analyte, a biosensor has got a bioreceptor. We've seen examples of bioreceptors such as your enzymes, we've seen examples of antibodies, we've seen examples of aptamers, and we've seen examples, the other examples I'll be giving you as we progress would involve molecular imprinted polymers or plastic antibodies. Remember this term, plastic antibodies, a very, very useful term because it's a very versatile technology, and this is going to be the future for tomorrow as well. Okay, so biosensor is a device which involves where the recognition system utilizes a biochemical mechanism. That's the keyword here. You have a biochemical mechanism here. We take a step back and look at where do biosensors find applications. What type of samples are we looking at, right? I mean, I know we have a mix in the audience. Some of you working in the area of environmental sciences, some of you working in the area of biology, of cells and things like that. So what are the different types of samples you can have? So if you look at electrochemical biosensors, which areas would your electrochemical biosensors fit in? First would be cell cultures. Anybody here doing cell cultures? No? I can give you, okay, yes, we've got Professor Khalid doing some cell culture here. I can give you some classic examples of human umbilical cord vein endothelial cells. These are cells I was working on, and we needed to know exactly. So when you have a cell culture, especially when you're trying to develop, look, the application area could be anything ranging from a diagnostic application to an area of developing a drug. Anybody heard about HeLa cells? Yes. Right? Has anybody watched the movie HeLa cells? The lady after HeLa cells. What's the specialty about HeLa cells? They're the first line of cancer cells which stay alive outside the body. And there's a beautiful program, there's a book actually, it's called The Immortal Life of Henrietta Lacks. Well done. That's, whoever has watched it, it's fantastic. It's a very good program, very good book to read as well. Right? HeLa cells, right? You have your HeLa cells, you're subjecting your HeLa cells to different forms of therapy in the process of developing cancer cure. Look, I'm telling you about biosensing, I'm telling you about wearable technologies. These are enabling technologies. I'm not asking you to take over the baton and start working in the area of wearable technologies or start developing wearable technologies. You could be working in another field and you can use these enabling technologies to gather more information. For example, HeLa cells. We take HeLa cells, we subject it to a therapy, to a chemical intervention. We subject it to a pro-apoptotic drug and an anti-apoptotic drug, and then we look at its expression in the form of signals that we're getting, and we need to monitor that continuously. What would you use there? Something which is a real-time wearable device that can be embedded in your cell culture, and it can give you concentrations of things ranging from oxygen levels. What are the oxygen levels? Are the cells under any form of stress? We know that lactate is a biomarker of stress. You can monitor lactate. You can monitor multiple analytes. So that's the beauty of technologies like wearable technologies and biosensing technologies. You do not have to be working in these areas, but you can take these technologies and apply them into different areas. And actually that's what we are doing, and that's what's called translational research. You take the technology, apply it to find solutions for human health care. Affordable, low-cost solutions. So this is the example of cell culture. Your sample matrices could be a cell culture device, wherein a cell culture petri dish, for example, where you have your cells growing. You could have human samples, right? People know about the pathology labs. You go to get blood tests done. You can get urine tests done. People go for fecal testing as well. So you can have human samples. You can have biological matrices. You can have plasma, serum. You can have urine. You can have saliva. You can have different forms. You can have sweat that we saw this morning. So these are your sample matrices, right? So this would be in the category of what we call as sample matrices. These are your sample matrices. Similarly, there might be anybody working on food technology? No? You could have food samples. right? You have food is being, food security is a major challenge, right? If you look at the major challenges that the world is facing, what are they? Energy, food and health. These are the three major channels, three major challenges that the world is facing. You could have food that's been exported and you want to look at the quality of the food. You have mangoes being exported from India all over the world. Are the mangoes still good enough? Because they're never, plucked fresh and exported out. They're always, they always are kind of picked from the gardens or picked from the tree when they're quite unripe, and then they're sent through. Are they still in a phase where they have not been infected by microbes, right? You need to get some answers there. So that's where another area where you can have food samples here, and similarly food adulteration, right? You all heard about plastic rice. You've all heard about cottage cheese, Indian cottage cheese called paneer being sold in the form of with all different mixed things. Milk adulteration, big problem, right? A lot of poor people, in the whole process of getting poor cows to milk more, they give them injections of oxytocin. Oxytocin helps in improving or increasing the productivity of milk, but it's a harmful chemical which, when ingested by humans, can actually have harmful effects, including organ failure. So you need some form of testing there. So that's where you have your food samples. That's your sample matrix here. And then you can have environmental samples. You can look into the environmental samples. You have the air, you have water, you have aerosols, you have soil, you have vegetation, you have wastewater plants, right? You can get a lot more information by just monitoring these. And for what do you need for that? You need biosensing technologies, right? So on the left hand side, this is your sample matrices, and then on the right hand side you see are your elements of biosensors. What do you have in your elements of biosensors? You, as I said, you have two systems which are actually combined in series. You have the bioreceptors and you have your electrical interfaces which help you in electrochemical transduction. Now this slide is mainly for electrochemical signal transduction. There's nothing optical here, so we are focusing more on electrochemical biosensing here. So what are the bioreceptors you can have? We spoke about APTA sensors, nucleic acids, single strands or double strands of nucleic acid which can bind very specifically to certain target molecules. We have your cells, cell-based sensors, cell-based biosensing. It's a new field, but it's definitely evolving. It's not evolving at a rate that we would like it to grow, but it's still there. And then you have your antibodies, which this morning we referred to them as immunosensors, right? Again, when we study biology in school, we learn about antigen-antibody interactions because we know that that's a classic example of what we call as specificity. Similarly, during our school days, if you remember your biology lessons, you learned about enzymes. And we would always start off by learning enzymes are proteinaceous in nature, and the nice thing about enzymes are you learn the lock and key hypothesis, and that's where you understand what specificity is. You cannot open any lock with any key. You have a key to, you have a specific key to a lock. So that's where your enzymes fit in nicely. Your enzymes gives it the specificity that you require to be able to target your target biomolecule or your biomarker of interest, so that you can get information just relevant to that. If I'm testing my blood for glucose only, I don't want to know how much lactate I have. I don't want to know how much acetaminophen I have. I don't want to know anything about the endogenous products and the exogenous products. When I say endogenous and exogenous products, I'm talking about exogenous means something that comes from outside. Endogenous means what is my body metabolizing and dumping into my bloodstream, which is then going into my cells. That's the endogenous products. Uric acid, ascorbic acid, these are all examples of endogenous products. Electrochemical biosensing, from electrochemical biosensing point of view, they are very relevant because they have their own electrochemical significance. What that means is that if you don't address the interference, you can have false values or false signals. Okay, so that's these are your what we call as the bioreceptors, and the next to these are your devices or your electrical interfaces which could be used for signal transduction. You could have potentiometric devices, FET devices. Anybody heard about FET devices here? Stands for field effect transistors. You can have ion selective field effect transistor devices. Basically, you're looking at something where you have different your different systems there, and you normally do a potentiometric measurement there. You can have nanowire arrays. Yeah, nanowire arrays, they're very interesting because they have a high surface area to volume ratio, right? And that's where the nanowire arrays are very, very useful. You could have nanoparticles. We've all heard about gold nanoparticles being used for optical detection. Gold nanoparticles are also being used to decorate the interaction and enhance the signal as well. People are these days combining receptors with nanomaterial systems. That way you can enhance the sensitivity. So far the examples we've been looking at concentrations where the requirement is measuring in millimole concentrations. You might have to go down to picograms per milliliter concentration or nanomole concentration. That's where the real challenges are, ultra sensitive detection. How do you achieve that? You have to combine your bioreceptors either together, two different bioreceptors, or you have to combine your bioreceptors with your nanoparticle systems to get the properties of a nanoparticle system, where you enhance the surface area to volume ratio and get a much more denser functionalized surface there. Okay, and then after this you have the next part, which is your electronic system, wherein you have a signal amplifier, you have a signal processor, and you have a display. I think these are things that will be covered by Professor Khalid in the subsequent lectures, but I think I'm going to stick mainly to these parts, which is your elements of biosensors, mainly to your transducers here. One of the interesting examples that we have in biosensors is glucose biosensors. I already mentioned this morning, it occupies a large chunk of the biosensing market, right? And I think if I have to teach biosensing, I normally give the example of glucose biosensing. Especially this is fantastic if you want to learn about enzymatic electrochemical biosensors or enzymatic biosensors. I'm going to explain how it works, and I'll do my best to explain it, and we'll take it from there, and we'll have a look at, the classic example would be that of glucose biosensors, which I say occupies around 85% of the total diagnostics market, and it's very, very important. Okay. Now, glucose biosensors has got a history, right? And this is also a way I can tell you a story of how devices evolve from first generation to second generation to third generation, right? We are all very lucky that we have a wealth of information to learn from. The first generation of glucose sensors were proposed and generated by Leland C. Clark. Unfortunately, this gentleman died in 2005. I think this should have been a person who should have got a Nobel Prize for his discoveries related to the biosensing principles and giving us the glucose biosensors. But anyway, that's a different world. I think he was American, a citizen, probably not very well connected to the institutions in Sweden, so probably might have lost out there. Anyway, I'm not going to go into that politics of Nobel Prizes. I'm just going to focus on the glucose sensors here. Let's have a look at glucose sensors. Okay, what do you think in a glucose sensor will be the molecular recognition element? What is going to be the bioreceptor? What is the best bioreceptor that you have heard of or know of, which will give the glucose biosensor its specificity towards glucose? Glucose oxidase, right? That's your glucose oxidase here, right? That's your glucose oxidase. This part is your glucose oxidase. Okay, let's see what happens. Where do you find glucose oxidase? Does anybody know where do we get our glucose oxidase from? Yes, please. I think you've given the right answer. Did you say fungus? Okay, the answer is fungus. You get your glucose oxidase from fungus. Glucose oxidase is produced from fungus. What's the role of glucose oxidase in fungus? Fungus's defense mechanism involves, every time it's attacked, the glucose oxidase helps in the production of hydrogen peroxide, H2O2. And hydrogen peroxide is the defense mechanism of fungus. It helps it kill the microbes that attack it. So the glucose oxidase is what is responsible for producing hydrogen peroxide in response to glucose. So we've done a very clever thing. In fact, this gentleman, Leland C. Clark, has done a very clever thing. Most of the things we do in real life also, we borrow those principles from biology or from biological mechanisms. I think Professor Khalid Mirza will tell you, the lab he was working from before was called the Bio-Inspired Technologies Lab. Technologies that are inspired from biology. Glucose oxidase or the glucose sensor is actually very much something that we have actually borrowed from the principles, working principles, from how the glucose oxidase functions in fungus or in fungi. What happens here? Don't worry about the cycles here. I'll explain it to you in a nice way. Just remember the few things here. This is your cofactor, right? This is your glucose oxidase. I'll represent it with GOX. Most of the people represent glucose oxidase with GOX. Glucose oxidase is an enzyme. Enzymes also normally have two parts, right? One is a prosthetic part and one is a cofactor. What's the property of the cofactor? Anybody? Any biologists in the room? Activating. How do they activate? how would you describe the chemical property? What if I said they are, yes, please go on.
By the help of ions?
By the help of?
Ions.
Ions, okay, yes, that's, you're marginally right there. Can I have a more accurate term to describe the behavior of redox species? Oh, I've given you the answer. The behavior of cofactor? They are mostly redox in nature. When you say something is redox in nature, that means it's something that can be reduced and oxidized. So you have your cofactor here, and there are different types of cofactors here. There's the FAD. Anybody knows what is FAD? Yes, flavin, and then adenosine dinucleotide, right? So you have your FAD, and then you have NAD, which is nicotine, right? Or exactly. And then you have your PQQ. You don't have to remember these names. As long as you remember GOX, that's fine. And remember, GOX has got a cofactor which could have one of these groups, FAD group, NAD group, or PQQ. PQQ are basically quinolones. Anybody worked on quinolones before? They are also redox molecules, right? When you say redox molecules, they are molecules which basically can be oxidized and reduced. Okay. Let's start now. What happens here? So is that clear to everybody? Your enzyme has got a cofactor which is basically by nature a redox moiety or a redox molecule, right? That's attached to your enzyme. You have your glucose which is present in your sample matrix, and this sample matrix could be anything. It could be blood, interstitial fluid, saliva, sweat, whatever it is. Glucose in that species diffuses from the sample matrix onto the sensor surface. The sensor could be anything. It could be a point of care screen printed electrode, or it could be a wearable sensor, an epidermal wearable sensor, or a subdermal wearable sensor. Doesn't matter. We're just looking at the fundamentals here, right? You have a metal surface there to which has been functionalized into a glucose sensor by attaching glucose oxidase molecules there. Glucose from the sample matrix diffuses onto the sensor surface, and you have your glucose oxidase. Glucose oxidase falls in the family of reductooxidases. What it will do is it will oxidize your glucose into a glucose lactone, right? The lactone will diffuse away. Glucose comes, oxidizes. So what has happened? A part of your cofactor here, your FAD, NAD, or PQQ, which is in the oxidized state, oxidizes glucose to glucolactone, but in the whole process itself gets reduced. That's what happens in redox reactions, right? Does everybody agree with me on that? That's what happens in redox. So this has now been reduced, right? And this reduced form of the cofactor then reacts with the oxygen that's present. If you're talking about biological sample matrices, blood, does it have oxygen in it? It has. Skin interstitial fluid, capillary blood, all of them have oxygen in it. So this reduced form reacts with the oxygen that's here and this reduced form of the cofactor reacts with the oxygen here and goes back into its oxidized form. So you have a cycle there. What is the advantage of this cyclic process? You do not need to it regenerates itself. Fantastic, very good answer. It regenerates itself, which means that you do not have to replenish the enzyme, right? And then what happens is when your cofactor reacts with the oxygen, it produces hydrogen peroxide. What was happening in fungus? Same thing. You were producing hydrogen peroxide there. This hydrogen peroxide, if you apply a potential ranging from 1 volt, 0.9 volts, 0.7 volts, would then break down into water and oxygen again. You again regenerated your oxygen here. And in the whole process, release electrons, which can be measured using your metal surface electrical interface, and can be seen in the form of an external current in your circuit using your signal amplifiers. All clear? Do you want me to repeat it? No? Everybody okay with me? Everybody with me? Still awake? Well done. Okay. If everything was fine here, do we need a second generation? So I'll repeat. Glucose gets oxidized, lactone goes away, the cofactor gets reduced, reacts with oxygen, produces hydrogen peroxide, and this hydrogen peroxide at 0.7 volts or 0.9 volts breaks down into water and oxygen and releases electrons, which can be measured in the external circuit in the form of a current. Yeah. How can you measure that in the form of a current? That's by instrumentation. We will be having special lectures on that, which will be given by Professor Khalid. This is clear? So if this is clear, and do you see any problems here? Does anybody identify any problems here? Can you see any problem here? Because that begs the question, if everything was fine here, why do we need a second generation? Pardon? Production of hydrogen peroxide? Yes. What's the problem with that?
So
It's corrosive. That's one good thing. Yes. What else?
So to detect, we need current generation, and electrons are getting, I guess, used up for the hydrogen peroxide generation. So I don't know.
Okay. Good try. Good try. I know what you're trying to say. You're trying to talk. You're actually trying to tell us about. You're alluding towards this high potential that we are applying, right?
Yeah. So that's why we need, I guess, second generation of electrons.
Okay. We'll come to that. Anybody else? Good answers, both of you. Well done. Well done. Anybody else wants to give it a try?
What about if there is some interfering agent which has similar redox potential as glucose?
That was the answer I was looking for. Here, the biggest problem here is interference. If you remember, when I was giving you introductions, I gave a lot of stress on endo and exogenous products. The body produces a lot of endogenous products, ascorbic acid, uric acid, yeah, and these are all electrochemically active. At this high potential of 0.7 to 0.9 volts, there is every possibility that you will have interference, because at this potential you can oxidize those other electrochemically active species as well. Plus, don't forget, we are talking about people who are regularly consuming paracetamol, ibuprofen and things like that. That's giving you your exogenous products. That's giving you your acetaminophen. And they can also interfere at 0.7 to 0.9 volts. So what do we need to do? We need to reduce our working potential. But if you reduce the potential, then as the gentleman pointed out there, sorry, what's your name? Vishal. As Vishal pointed out, you wouldn't get the required sensitivity there, because low potentials will give you low currents. You wouldn't get the required sensitivity there. So therefore, we actually need to reduce this. So to come across this problem, in 1984, at the famous Oxford lab, three gentlemen received a blue plaque for this work. This was done in 1984. This is Professor Tony Cass, my guru as well. Hill and Davis in 1984, they did some pioneering work wherein they showed that glucose oxidase enzyme reacts very well with certain chemical species which are also known as mediators, ferrocene based mediators, ferrocene derivatives, which are basically iron atoms sandwiched between two cyclic rings, right? Those are called as ferrocene derivatives or ferrocene compounds. Ferrocene compounds are also redox mediators, right? Because you have iron there which can be oxidized and reduced from Fe2 to Fe3 and Fe3 to Fe2. Yeah, you can oxidize Fe2 to Fe3 and reduce Fe3 to Fe2 plus as well, right? So you have those ferrocene mediators there. So Professor Tony Cass, who is also my guru, and I also like to mention here, he was your Professor Khalid Mirza's PhD Viva examiner as well. He was involved in giving this pioneering work that was given in 1984, which is also the basic principle that is the underpinning technology that has given you the modern day glucometers. How many of you have seen glucometers? Well, almost everybody has seen glucometers. If you have anybody, whether it's a grandparent or an uncle or anybody in the family who's trying to keep their glucose levels under control, you would see them using the glucometers. I mean, it's now become very, very common, no matter whichever party, whether you go to the most advanced cities or whether you go to the remotest part in the country, glucometers have become very, very common these days. Okay, so this is basically the second generation. What happens in second generation? You actually are having a cofactor, and this cofactor doesn't interact here. So the first step, so this is the first step. The first step says the same. In the second step, you bring in your mediator, which takes over the oxygen concentrations. right? Don't forget, when tissues are sick or tissues are not well, unwell tissues will have a varying concentration of oxygen. The oxygen levels drop down as well. So measuring, especially measuring glucose in high altitude is always going to be a problem if you're using first generation, because oxygen levels are low there. And if you don't complete the third step, the second step here, then you cannot get good glucose sensing. You can't get accurate glucose sensing here, right? So you have your mediator here, and this mediator basically, then what you have, this cofactor then interacting with this mediator, and this is the whole basis. This is the whole NMR work that was done by Professor Tony Cass during his tenure at Oxford in 1984, for which he's been awarded the blue plaque with Hill and Davis by the Royal Society of Chemistry. Blue plaques are very, very prestigious awards. They're normally given to people after they're dead, but these are, I think, probably the first 3 people who are alive to receive a blue plaque, and I think which is a good decision as well. All right. So you've started using mediators here, and this is how your second generation happens, right? So in second generation, you have glucose diffusing in, getting oxidized by the cofactor, and then the cofactor will transfer that electron, or there's this electron transfer with the mediator, and the mediator will oxidize it back to its oxidized state and get itself reduced, and then you apply a potential. And in this particular case, you can apply anything between 0.4 to 0.1 volts. At low, I think 0.4 to 0.2 is normally the range volts, and that will then give you your good signals there, because at that particular potential, these mediators are very much active, right? You don't need very high potentials there. So you can change the over potential range and come down, bring the potential lower, and you can also cut down on the possible interferences. Okay, so that's the story of second generation. Is that clear to everybody, or do you want me to repeat it? Is that all clear? All right. So if the second generation solves all the problems, why do we need a third generation? Pardon? For electron transfer. I know you've read it from here, direct electron transfer. That's fine, that's a good try. Why do you need direct electron transfer? What's wrong with this one here? Pardon? Speed.
Speed, rate.
These things work at very, very fast speeds. Electronic shiftovers are, that jump, electron transfers are at the same speed as electromagnetic radiations.
Mediator specificity, I think.
Mediator?
Specificity.
No, the mediators are well established mediators. Any biology students here?
What about the location, the active site of the enzyme?
That's all very well found. I mean, look, there's been a technology which has been here for ages now. So think about it. When I'm giving you some clues here. You have the glucometer technology, very well tried and tested. I mean, people still can't find faults in it. They're still using it for managing glucose, and it's doing a great job. But the requirement has changed. What is the requirement now? What is the requirement now? Cost? Yeah. What else?
Extra energy will be used, which we can cut down in this directly.
That's from the, that's okay, fine, I'll take that one as well. Exactly. You're moving away from point of care to wearables. So you want something which is more continuous monitoring, right? In the morning we did see, right, that when you do measurements with glucometers, you have point measurements. What we want is a full profile of how the glucose concentration changes there. So what's the problem there with wearables? What's the problem there with wearables? Why not?
Because there will be no direct contact of the
The slides are jumping. Yes, sorry, one more time.
So there will be no direct contact of this cofactor with the electrodes.
Why not?
For if we are wearing this wearable things, so there will so if we will go for the beginning, as like we take out the blood and then we measure the glucose level, because there will be a chemical reaction, and
Yeah.
that will be helping in measuring the currents. So here we don't expect that chemical reaction between, there will be no like mediators and all. So wearable things, they should not penetrate the things inside the like body, and they cannot take the things.
You're giving, I know what you're thinking. So you're talking about biocompatibility.
Yes, biocompatibility.
So your mediators here, are they biocompatible? Are ferrocene derivatives biocompatible? Do you think ferrocene can sit under the skin? In a subcutaneously implanted sensor, if you look at it, it's not like a thin wire which has been functionalized with the enzyme, right? Does it sit in the, when it sits in the skin, it comes in contact with the underlying tissue. Biofouling is there. Biofouling is an immune response. We'll come to that as well. Are ferrocene mediators biocompatible? The answer is there's a big question mark there. Most of them are not, right? And therefore, there was this thought process of coming up with something which allows direct electron transfer. What most of you said, that's all right, true as well. You need something which is faster, you need faster direct electron transfer. But again, you need to understand, if you look at the scale of things, right, you have the surface and you have a molecule standing up right there. You can have direct electron transfer from the parts which are lower down. You cannot have direct electron transfer from the upper part, right? So you need to design, or you need to engineer enzymes, modify existing enzymes so that they have structures which are involved in direct electron transfer. And this is some work which has been pioneered by Professor Koji Sode, who is one of my collaborators from Tokyo University of Agriculture and Technology in Japan, right? Now I've mentioned about biocompatibility. If you look into, or if you do research on the Abbott biosensors that are available for real-time continuous glucose monitoring, they are actually based on osmium complexes, right? And osmium complexes are basically mediators. Yeah, there you go. So that's the whole story of how the generations evolve from first generation to the second generation to the third generation. In the end, I'll give you some time to ask any questions that you may have on this. In the interest of time, let's move on to the next slide. Yeah. Okay. Now, just taking a step back, a little bit of a reminder of our early days at school when we learned about electrochemistry. What is electrochemistry? Well, we all know electrochemistry is the study of chemical processes which are involved in electron transfer, right? You have fundamentals of electrochemistry being applied in different areas. It includes batteries. These days, there's a lot of work on batteries. People are talking about having battery-driven cars. So there's a lot being used in lithium carbide, lithium batteries and stuff like that, lithium battery technology. There's a lot of electrochemistry that is involved in corrosion studies, because corrosion at the end of the day is a oxidation process, right? So you have a lot of, you do a lot of electrochemical studies or applications in corrosion studies. Sensors as well, you need a lot to understand a lot of electrochemistry. There's a lot of electrochemistry involved there, and of course biosensors. Now the basis of electrochemical biosensors mainly involves measuring redox active analytes, right? If you look at biosensors, why is electrochemistry important there? We are mainly looking at the redox. I've already given you an example of glucose oxidase there, where I showed you how the cofactor is involved in a redox process there. It's getting oxidized and reduced, and that's where the electrochemistry comes in. Let's have a look at some of the other things as well. This is an example of how you would modify a wearable device with lactate oxidase. I gave you the example of third generation. This is an example of L-lactate. This is a third generation lactate oxidase sensor. And what we have here is, if you look at the way we use it in a wearable format, you would have arrays, multiple arrays, right? If you remember in the morning I showed you there were 4 islands, right? Those islands have got a purpose. Each island acts as a single electrode. And what do you have there? So this is represented, the reference electrode is represented as a single electrode. You have a counter electrode, and then you have your working electrodes. And this dot thing that you see is basically your bioreceptor. It could, in the case of lactate oxidase, it would be lactate oxidase, which is actually entrapped in a film of polyphenols. And what we do is we normally do electropolymerization. Electropolymerization limits the film dimension to 50 nanometers. It's very thin and it's very conformal. And it's like a thin film which has got the enzyme entrapped. You don't want a very thick film, right? Because if you have a thick film and your enzyme is sitting down at the base, it cannot, the molecule cannot diffuse down to it. So you need a thin film, and you need the enzyme redox sites to be very, very accessible, right? So therefore you use things like electropolymerization, things that give you very thin films, so that you can have a very effective ones. Otherwise it's your sensor becomes restricted by mass transport. There's not enough mass transport to the sensor surface, and you then end up getting very low values of current and things like that. Just a quick summary on the redox reactions for those of you who, just as a refresher, we all know oxidation is loss of electrons, reduction is gain of electrons, and you all normally have a redox pairs. You will have glucose, which will give you glucolactone, which will release 2 electrons, and you'll also have 2 protons being formed. So you always have them in pairs, and if you look at the sum total, it's always the same. Charge and mass normally is conserved, nothing is lost. This is the law of conservation of charge and law of conservation of mass. And the core to biosensing signals this is also the core to biosensing signals in your enzymatic electrochemical sensors that you normally come across. What are the different electrochemical cell components? People who have not done electrochemical cells or worked on biosensors, if you look, then you normally see, if I draw you here, you have a central electrode, and then you have something around it, and then you also have a third electrode which can so you can have something which is, this is continuous, and then you can have a third one here like this, right? So this is going to be your working electrode, this is going to be your counter electrode, and this is going to be your reference electrode, right? This is where your sample drops in. Your sample sits in the form of a drop. So this is like what you would normally see in a strip, and these lines are actually screen printed lines or electroplated lines if it's a PCB, depending on how you've metallized it. So these are metal lines. These could be anything ranging from carbon inks to gold surfaces to platinum surfaces, and your reference normally is a silver chloride reference electrode, and then you will normally have a pad here, another pad here, another pad here, and then this will form the part of a connector, and it fits into a connector, and that connector will then make connections to a potentiostat, right? That's how it normally works. So your working electrode, this one here, is the site of redox reaction. This is where the reaction takes place. Your reference electrode is there to maintain constant potential, and you normally can have different materials. I don't know if I've got that in my next slides, but we'll have a look. You can have, for your working electrodes, the typical materials could be ranging from carbon paste, you could have gold, you can also have platinum, and then you can have different types of materials there, right? These days people have come up with P dot and different types of conducting materials as well. You can also have organic materials as well which are conducting. You can also have organic electrochemical transducers or transistors. Yeah, so these are different materials that can be used for your working electrode. It depends on what your application area is. You need to have a stable electrode. So somebody mentioned about using gold electrodes. When you use gold electrodes, when I started off my work with wearable sensors, we had some people who had worked on gold electrodes, but we forgot to take into account that gold electrodes work very well in a second generation format, but it doesn't work very well in the first generation format. Because what happens is your baseline starts for gold here quite high, and if you move to platinum, then you have a very low baseline, right? So then when you do minor additions, you add 0.5 millimole, you get a step. Then you add 1 millimole, you get a step. You add 2.5, you get another step, and you get step like this. While with the gold, so this is your 0.5 millimole, 1, 2, and 5, and then so in the case of gold, you don't see anything when you add these 3 additions, and you start off seeing things at 5 millimoles because your background is quite high, right? And it's very important that you work with materials that give you low background currents and stable background currents, so that there's less drift. So when you start, it goes down, and then it stays stable over a certain period of time, right? Again, there are electrochemical principles there, but if I start using those terms here now, it might scare you guys. So I'm just going to stick to what's relevant to biosensors. Okay, so you have a working electrode, as I said, carbon, gold, platinum. You have a reference electrode, which normally you can also use if you want. Some people do use screen printed electrodes where you have a gold working electrode and a gold reference electrode, but that's not very stable. If you want to do long term measurements, you need to use a silver chloride represented as this. It's very easy to make them. You take silver, you sputter silver, have a thick coating, and then treat it with ferric chloride, or you treat it with bleach and you can chloridize the surface and you can get silver converted to silver chloride. Yeah, and then you have a counter electrode which is normally used for completing the circuit. The thing you need to be careful with the counter electrode is the surface area of the counter electrode. Apparently the thumb rule is that your counter electrode should have twice the area of your working electrode. These are things that go into your device design. And then finally you have an electrolyte. The electrolyte sits on top of this and it gives you what the connectivity there, and it's this electrolyte which is your sample matrix as well, right. It's from this electrolyte that your sample will diffuse towards your working electrode, and you do all the measurements on the working electrode. You apply a potential with reference to your reference electrode, and the potential is applied between your counter electrode and your working electrode. That's how the whole thing works. Now we are going to be looking at different techniques that are normally used, right. What are the different electrochemical techniques that can be used? You can use potentiometry. What is potentiometry? When you are measuring the potential difference between your working electrode and reference electrode, that's called as potentiometry. You are basically looking at changes in your potential. There is no current that will flow. Examples for these would be ion selective electrodes. I think these are something that's some work that's already being done at NIT Rourkela, wherein you have your you have your chromophores, right, your ionophores. They have ionophores. Ionophores are the ones that give specificity. So if it's a potassium, it's the size, it's got cavities that are the size of a potassium. So your ions, what will happen is you have a working electrode. On the working electrode, so you have a working you have your working electrode. On your working electrode you will have a layer of your ionophores which will have cavities on it, right. And what will happen is, as soon as you have your ions which are of different sizes, so these cavities are specific to potassium, specific to sodium. So if you have potassium ions, if they are specific to potassium ions, it will come and bind, but sodium will not, right. And then you measure the changes in using ion selective electrodes by applying a voltage, by measuring the difference in potential between the working electrode and the reference electrode here. Okay, that's how you measure potential. That's what a potentiometry is. I mean, everything is in the name here. Potentiometry. Metry means measure, right. So you're measuring changes in the potentials, and that's a good chemical signature. That's a good signature of a chemical signature of a compound or a biomarker of interest. This is an example of how a subdermal variable device has been modified into a potentiometric device. This work was actually published in Electrochemical Communications. This was the first proof of concept studies that were done back in 2017. As you can see, we have 4 different islands here. Yeah, and these islands are meant to be working as your working electrodes. They are made up of gold. You have a silver here. The silver is a silver sputtered island here. That is normally metallized. That is normally modified to a silver chloride reference electrode, right. And these pyramid shaped structures are your microneedles, right. You modify them with, first of all you have a gold layer, and then the gold layer, and then you have a layer of iridium oxide. Iridium oxide is good in looking at pH changes, and on top of that you put an enzyme called as beta lactamase. And what is happening is, when penicillin, and then you put a hydrogel here for tissue compatibility, because you are sticking these devices to the skin, you don't want any adverse reactions there, so you have a very thin layer of the hydrogel there. So you are having penicillin, which is a drug, right. This drug is going to diffuse through the hydrogel layer onto the iridium oxide and onto the gold, and you will normally see changes in the pH, which is normally measured in the form of a potentiometric sensor. I mentioned that potentiometric sensors are also ion selective electrodes, and when you say pH, you're basically looking at H positive ions, right. So you're generating protons here when penicillin is oxidized by beta lactamase or beta lactam oxidase to penicillinic acid here, right. This is a very important application. Why do we need to monitor penicillin in the skin? Anybody here? Can you guess the application here? What is penicillin? Can you think of a big problem with antibiotics?
Sir, resistance.
Well done. Yeah. AMR, antimicrobial resistance. Well done, right. So what does it mean? Overuse or underuse of antibiotics leads exactly, you start to develop this thing. So this gentleman here, Tim Rawson, was a PhD student. He's basically a clinician who worked on this, and trust me, a clinician working in a lab doing biosensing experiments was a novelty there. This is the work culture there that we have in the UK, and this is how translational research is possible. For clinicians to come and work in chemistry labs, and for chemistry lab and engineering people to go and work in the clinic, that's how translational research happens. If you don't have that collaboration, research doesn't go out of the lab. That's what you need to understand. Okay? So here this was the whole idea of developing a penicillin sensor for therapeutic drug monitoring in the skin compartment. Right? So what happens is that you have penicillin, as I mentioned this morning about personalized medication, right? If we all admit, if you look at the way people, diseases are treated, there's a one size fits all kind of an approach, which is not a very good approach. And that's the problem with antimicrobials, that you start to have antimicrobial resistance. You can have resistance against antibacterials, antifungals, antivirals, right, by overuse. So you need to only take as much as you need. So one way to do that is by looking what we call as doing PK/PD studies, pharmacokinetics, pharmacodynamic studies. That helps in understanding how your body absorbs and reacts to the different medication there. If you understand that, it then helps in administering dosage according to your requirement. Most of the whole world practices prescription regimen wherein one size fits all, drugs are given to you according to your age, body height, body weight, and things like that. That's not how things work. Things work based on your own individual PK/PD values, how you absorb and metabolize drugs. Right? If you do therapeutic drug monitoring using wearable sensors in the skin, that gives you a good idea as to how much of the drug has been metabolized and how much of the drug is still available in the form of bioavailability. Right? That helps you calculate that, and that helps you in kind of tailor making therapies, which allows in prescribing drugs as well according to the person. This is another one wherein basically I am showing you some real measurements. As you can see, this is continuous measurements over a period of 6 hours. You have two different traces here. You have the different microneedles here, one which is basically used as a control, and then you have another one which you can see is giving you values which you do a good fit here, and then you see you are taking a penicillin here and you are getting a peak in response to this particular penicillin here. Just show you a little bit more on the instrumentation. This is your microneedle array, or the subdermal patch that we call it, with the wires there sticking out, and this is inserted on the hand. We were not using any applicators here, and then you are doing the measurements here, him sitting there. This is your potentiostat, which you will be learning more about and looking at the results there, and this is how we were doing the measurements over a period of 6 hours. What you can do is, if you want to find out how a person reacts to a drug, give it to the person and then look at this peak. For somebody this peak will be here, for somebody this peak will be here, for somebody this peak will be here. Right? What you need to know is what is the therapeutic index. So there is an index, there is a certain level in which the drugs will work. If the drugs are above this level, then you have harmful side effects. If the drug is within the range, perfectly fine. If it's below this, then it doesn't work. Right? So it's very important that you get this therapeutic range. Some drugs have got a very narrow therapeutic index or a therapeutic range. So what is the disadvantage there? They can easily, you can easily overdose and have deleterious effects. You can have side effects, right? That's the problem there. Okay, so this is an example of how in vitro monitoring, how we use potentiometry for in vitro monitoring. Okay. All right, amperometry. Somebody asked about chronoamperometry. Amperometry, as the name suggests, metry means to measure, ampere means current. Amperes is the unit of current. This is basically where you measure current, but for this you apply a constant potential. You don't vary the potential. You don't look at the changes in the potential. You're measuring the current in response to a fixed potential there. What's happening to the current there, right? Example here would be a standard enzyme catalyzed glucose sensors here. We've already seen this one here, and you have a signal that's directly proportional to the analyte concentration. So if you remember that, glucose oxidase in oxidized state and glucose oxidase in reduced state, right? And you had this glucose which diffuses, and you have this going from this state to this state, right, and here you have a lactone being produced, right, and then you have this whole thing again running, and in response of this you have your, for example, if you have oxygen, then you are generating hydrogen peroxide and you are getting a signal, right. So if you measure the current over time or concentration, your signal will directly vary with the concentration. Higher the concentration, higher is the current, right. So you are measuring this current which is directly proportional to the concentration of the analyte, right. Can you see how you are going backwards, but you are using this whole process to measure one thing which is your glucose very, very specifically, nothing to do with interferences there. Yeah, is it getting clear now how we are using amperometry now in real practice? And then you can also do continuous monitoring of other metabolites. We have examples of glucose biosensors. So there is a paper here which I published in Sensing and Biosensing Research. I have given you this paper because here we measured glucose sensor using glucose oxidase, lactate sensor using lactate oxidase. Lactate oxidase, this is with the membrane and one is without the membrane. These are kind of good examples here, and as you can see here what is happening here, you have this, so you do what is called as a dose response curve. Those of you who work on biosensors know this term very well. Dose response curve is where you measure what are the current values in response to the concentration increase here, right. So you are developing a sensor, right, you have got an enzyme, you have developed a sensor, you have got a new material, and you are looking at how my sensor is performing. So you do a dose response curve, and all these studies are part of what we call as in vitro studies, right. You take your sensor and you first of all just have a blank PBS solution. Why PBS? Because it is a physiological buffer. In most of the systems in the body, the physiological buffer conditions is it maintains a pH of 7.4 or 7, around between same range, and what is the ionic strength? It is in millimoles. So normally you have a PBS solution which ranges from 10 millimoles to 100 millimoles, and that is where you prepare a solution. That is your background. So you get your background value, right, and then you do values here. So this one here where shows that we did measurements for 0.5, 1, 2.5 and 5. So we get the values there, and based on this you get a dose response curve. Now what happens is this curve goes up and then at some point it starts to plateau, right, and this happens a lot in enzymatic systems, and that is because we say we are doing a Michaelis-Menten fit, because that explains enzymatic kinetics. So the point at which you have this linearity, half that point is called as a Km value, or the apparent Km, this thing that tells you, if you look at the Km value, you can then say, oh, my dynamic range is this much. So for example, if your sensor is giving you a response of this much, this is around 30 here, and then it plateaus, then the Km value will be around 15. That will be suitable for systems where you have very high fluctuations, where the values are going very high, right. Animals, very low dynamic range. So this range is called your dynamic range. The linearity range is called your dynamic range. What is the dynamic range? So you have to match your dynamic range of your working sensor to the range of the biomarker in the biofluid. If your sensor is capable of measuring from 0 to 30 nanomoles per liter and the dynamic range is in millimoles, it is no use, because you are far ahead of that. You need to match the two, dynamic range of your sensor's performance and the dynamic range of, in fact you design your sensors so that they give you a performance which matches the dynamic range of the biomarker or the analyte of interest. Yeah. So this is an example of glucose oxidase, this is the example of lactate oxidase. I think I also have theophylline as well. So we have also used here, I think in this case we used xanthine oxidase. Xanthine oxidase is used for monitoring theophylline. Why theophylline? Anybody here who works on drug chemistry understands theophylline? It is a bronchodilator, right. It is a bronchodilator which is again got a very narrow therapeutic index. So if it exceeds that level, if you give too much high concentrations of theophylline, it is then going to cause cytotoxicity. right. So we are measuring that here, and then this is an example of again the same paper wherein we've got a potentiometric sensor. You can see we've got a good range, but look at the difference here. This one is in concentration here, this one would be millimoles, and this one is in moles per, this is also moles per deciliter, but again concentration we have to match the dynamic range with that's seen in biofluids. Moving on to the next method, next electrochemical method, it's voltammetry. In voltammetry you are measuring current as a potential as you sweep the potential, right. So this is called as voltammetry, measuring current. So you might have heard about cyclic voltammetry. Anybody seen those birdie like structures with redox mediators? So we spoke a lot about redox mediators, right. So when you have a redox mediator like ferrocene carboxylic acid or any other redox mediator, when you apply a potential and you make a sweep, if this is your voltage and this is your current, you can see that when you apply an oxidation potential, you go from this to this. Let's say you're going from 0 to 0.6 volts. So you go up, you get a oxidation curve peak, then come down, and then you get a reduction peak. It looks like this, right. This is your EOX, this is your ERD, right. And normally for a standard case, the Nernst potential is the difference between EOX and E reduced. Ideally it should be, anybody, Nernst potential? Yes, the ideal would be 0.075 or something like that, but normally when it comes to chronoamperometry and things like that, where you are not doing very sensitive measurements, you accept 0.1 to 0.15, right. And this, what does it depend on? The peak position. This also depends on your reference electrode. Yeah, it's also an indicator of how stable your reference electrode is. If you do, normally when you do cyclic voltammetry, you don't get one trace, you do multiple sweeps. So you start the first one is usually quite a big one, and then comes down, and then you have a second one which is then smaller than this. If it looks like this is not reproducible. A reproducible one would be something which gives you this, comes down, and the next one is very close to this and comes down, literally overlapping. That tells you about the stability of your working electrode surface as well, and the peak position tells you about your stability of your reference electrode. If the peak position is shifting, that means your reference electrode is not stable, it's moving. Yeah, that's how you normally use this voltammetry techniques. So you have cyclic voltammetry, and then you have another technique which is called as differential pulse voltammetry. Differential pulse voltammetry is where you apply pulses of, and you measure look at that, you do a differential subtraction there. And then you have square wave voltammetry, wherein you basically apply voltage and you measure versus time as well, and these are very useful for looking at redox active compounds or label based sensors as well. Some examples here, this is some example from my research group. These are, as you can see, current versus potential measurements versus a pseudo reference silver chloride here. You can see here you are getting this peak here, and then this peak starts going downwards. Why is it happening? Because in my group we work with something called as molecular imprinted polymers, right. What you have is in molecular imprinted polymers, you actually have a working electrode. On the working electrode you have a polymer solution which has got your target biomarkers, right. What you do is you do electropolymerization to solidify it, and you get a very thin layer which looks like this. A very thin layer. I will just magnify this thin layer, and this is your target analyte molecule, right. And then in the next step you bring in oxalic acid and you eat away this whole thing, and you create cavities here. These cavities are very similar in shape, size and charge to your target molecule. So when you have your target molecule coming in here, it would come and bind there. Now what happens is, as when you have nothing, you can see that there is a gap here between this layer and there is a good electron transfer here, right. When you fill this gap up with the target molecules, as the concentration increases, you keep on blocking the gaps. That will then change its electrochemical properties, that will change the impedance levels, and that then shows us in the form of a drop in the current values. So you can see here this value is for 0 picograms per milliliter concentration, and this value here is for 22.5 picograms per milliliter concentration, right. Pay attention to the fact that we are measuring in picograms per milliliter concentration, which is basically looking at nanomolar concentration, right. So that's low concentrations, and this is the work that we were doing basically to look at biomarkers of Alzheimer's disease, p-tau proteins in blood plasma and serum samples and all those etc. Okay. So these are examples of DPV or differential pulse voltammetry, and then another technique is called as electrical impedance spectroscopy. In electrical impedance spectroscopy, what you do is you measure impedance over a frequency range, right, and then what you do is you have, if you look at the typical Nyquist plots, you will have a non-Faradic and a Faradic process there, which will basically be represented in the form of the semicircle followed by a line there, right. This is again a label free and a sensitive method to look at surface binding here. Same principle here, molecular imprinted polymers, you are looking at the changes in the impedance there, you are looking at the changes in the electrochemical properties. This is electrical impedance spectroscopy here. You can use this for bioreceptors such as aptamers, and you can also use them for antibodies, including plastic antibodies, the one that I described in the last slide. This is an example where we have used electrical impedance spectroscopy to look at for subdermal variables for IL-6. Does anybody know what is IL-6? Yes, cytokines, right. IL-6 are cytokines. So what did we do here? We took our microneedle array, right, and we created cavities here. Sorry, we took a solution here with your biomarkers that you need, and we electropolymerized it, and we created a film which has got your biomarkers entrapped in the film. In the next step, this is the next step, which is template removal. We remove these biomarkers using either proteinase K, which is an enzyme, or using oxalic acid. It will come and eat away the red dots there, leaving cavities there, so that when these microneedles come in contact with the solution of IL-6, the IL-6 will come and bind at these sites there, and that binding will be represented in the form of changes in electrical impedance spectroscopy. You can see here different EI Nyquist plots you are getting here. So you will get this for different concentrations here, and that is what we are measuring here, right. So this is an example here. This has also been published work from one of my own work. And the nice thing with electrical impedance measurements is when you are preparing the template and you are removing the template, all of this can be followed electrochemically very easily, right. If you have an insulating surface and you have a redox species on top of it, you will not be able to see any signals there until unless it is a very porous material there. But when you have a conducting surface, you will see clear oxidation and reduction peaks here, right. So you can use, this is an example where we have used cyclic voltammogram or cyclic voltammetry to look at the coating. Are we getting a nicely conducting surface, or are we getting an insulating surface? How is this, how is it changing? You see here, from here, the golden curve here, trace here, it is changing to this blue trace here, right. That is just showing you a drop, which shows that you are getting a good layer, because the insulating material that you have on top of it is mainly polyphenol, polypyrrole, and those kind of things, which are electropolymerized, right. And then you have your curves here. You see this is for the bare one. For the bare one, you are seeing a very small Nyquist plot, and then you have this one is where you have a molecular imprinted polymer, you have a much bigger Nyquist plot, and then when you remove the template, when you remove the biomarkers, the plot changes. So what I am trying to say here is we need electrochemical techniques to follow the whole process of building biosensors, right. Similarly, you have these graphs here and Nyquist plots here, which are showing you electrical impedance measurements. Now you would ask me the question that if you are using why did you use EIS here, and why are you using DPV in your current work? And then the answer to that is EIS can also destroy your structure. Sometimes sometimes some of your techniques are capable of damaging your surfaces, because you are looking at very thin films. You are looking at films which are like nanometers, tens of nanometers, maybe maximum 50 nanometers. for these very thin films, it's good to use these electrochemical techniques to monitor how things are growing and things like that, but for actual biosensing measurements, it's always desirable that you are able to reduce, you are able to move away from EIS and use more other techniques which are much more gentle and much more nicer to follow up or to look at the dose response curves. And then this brings us to the wearable biosensors, how we can integrate electrochemistry here. Electrochemical techniques can be miniaturized, right? I already showed you the example here of this one here. So you can see microneedle arrays. I also showed you the microneedle arrays which were functionalized with lactate oxidase, and I showed you how you had modified some of them into a working electrode, some of them into a counter electrode, some of them into a working electrode. So that's how you can use them. And if you have multiple electrodes, you see this picture here, if you zoom into this particular picture, you can see there are 4 islands. The idea is you have 2 working electrodes, 1 counter electrode, 1 reference electrode, right? Or you can have, if you want to do a 2 electrode system wherein you only have a working electrode and a counter electrode and a reference electrode, you can have 3 working electrodes and one of the other combined reference and counter electrode system there. And then there is some case examples that I think I have already given you. So on glucose and lactate sensing, I have also given you examples of potentiometric sensors for electrolytes and impedance based measurements. That's not me, right? I think that almost took us to the last slide, and I think with that we can probably, have I run over time?
It's okay. We can move on to Q&A.
Yeah, I think we can now open the floor for questions.
Yeah.
I only won't be able to take you to the slides unless it comes back on my screen.
Right. So, anyone, any questions?
Sir, can you repeat what you said about EIS on porous matrices? You mentioned something that the current will change if we do EIS on porous matrices.
Yes.
And the curve will change. Can you repeat that?
All right. So let's imagine. I can't use the screen right now.
Just give us 5 minutes. And
Yeah, no worries. Imagine you have a surface, and you have a surface which is coated with a polymer, and the polymer is basically a template for your biomarkers, right? And now imagine a situation wherein you are introducing your solution, which is like a blood sample, and the blood sample is sitting on top of on your sensor surface, and your biomarkers are diffusing towards the cavities that we have created, corresponding to the templates we've created corresponding to this. As they keep on binding over time, so when you have only because you have cavities, the impedance is there's low impedance there. As you keep on filling those cavities with your target molecules, you start to build more and more, you start to have more and more resistance and impedance, and that when you measure, you can actually see it in the form of the Nyquist plots.
So for paper based electrochemical sensor, is this becomes a very big problem because of this reason?
I mean, we use electrical, a lot of people are still using electrical impedance spectroscopy. I am just sharing the experience of my research group, that my research group prefers using differential pulse voltammetry when it comes to getting dose response curves and things like that, because they think that EIS is not very gentle to systems which have got nanoparticles and nanofilms. Because in EIS, don't forget, you're also having the frequencies, right? I think you're using a wide range of frequencies there as well, and sometimes that's not very gentle to the system that you're testing it against.
So we apply a signal, changing frequency signal.
Changing frequency as well.
So it might affect the surface chemistry. So that's why we go for DPV generally. Yeah. Any more questions, please? Please feel free to ask. I have a couple of questions.
Yes, please.
Sanjeev, just you briefly mentioned in the beginning about plastic antibody, and you mentioned that it is something quite interesting.
Yeah.
So can you please elaborate a little bit more on that plastic antibody? What are the potential applications and all that?
I think I'll give you a short answer now.
Yeah, short
But we
answers.
will have I think probably one of the case studies is dedicated to the plastic antibody, and for that I think actually also have some good animations there as well.
Okay.
So plastic antibodies is a simple thing. The term is called molecularly imprinted polymers, right? So I will go back to the slides where I actually have this one. This is a good example here. So what you have is, you have your microneedle array. On this microneedle array, you bring in a solution of a monomer, for example phenol, or you can bring in pyrrole, right?
Yeah.
And phenol can be easily electropolymerized to polyphenol. Pyrrole can be easily electropolymerized to polypyrrole. In this solution, you have your target and target biomarker. So you have the working electrode, and on this you form your thin layer, and this layer has also got your molecules, your target molecules are trapped in it, right? So what you do is, you then bring in oxalic acid solution and you remove this is called as, you remove the templates, you get rid of these, and in the process you end up having, if I show you a magnified view, end up having surfaces that look like this, right? So now in the next step, when you have your target molecule coming in, they will start coming and binding to these, and as they bind, you will see a change in the electrochemical properties.
So these cavities, are they have some logic behind the size of the cavity?
Very much, because what happens is, that's why I said I'll give you a short answer now and big answer will come later on, because the first step of the monomer, so what happens is, you have your monomer and then you have your target molecule. So before you start electropolymerizing it, the first step is called as interaction mechanism. That's a very, very important step. During that step, there is a lot of interaction between the molecules, so it will bind in a certain, it will bind to the monomer, your target molecule will bind to the monomer in a certain configuration, and there will be charges that will stabilize and destabilize it. So it's very important you tweak your polymer or your monomer properties to make sure that you capture your target analyte in its best form without compressing it, without changing anything, and you also capture it in a state so that not only do you get the corresponding size, because you can always have different molecules of the same size, but you also have the charge centers are also aligned in the same way, so that that charge center, that there is a binding not just because of the size but also because of the charge. That's what gives it the specificity.
Okay. Yeah,
Right?
very
And then
nice.
and then in the next step you remove this and you created cavities. So when the target analyte comes in, it will come and bind not just based on the size but also on the charge and on the interaction. You could have hydrogen bondings, you could have pi-pi interactions, because you are looking at cyclic compounds. There are different forms of interactions which will support the interaction and which will ensure that your cavities are in a form to which only your target molecule will bind, and on top of that you do interference studies. I might have a whole lecture on just on molecular imprinted polymers.
Thank you. Any more questions?
Yeah.
Want to know how thin you can make? You were saying 15 nanometers, something like that.
Yes, it's a very good question, and thank you for asking this question. I might have forgotten to mention this. So what happens is, phenol in solution is conducting, polyphenol is insulating.
Yeah.
So phenol, as soon as it starts to polymerize into polyphenol, it becomes self-limiting. It does not allow polyphenol to grow beyond 50 nanometers, and that's the typical size of the molecules of the biological molecules, whether they are macromolecules or small molecules. And therefore these plastic antibodies are actually have been developed not only for large molecules but also for small molecules like glucose. People have used MIPS for glucose sensing. People are using MIPS for iron sensing.
Yeah. So those polyphenols are like, if you have electrode, so polyphenols are lying this way or this way?
Doesn't matter.
Doesn't matter.
Doesn't matter. Doesn't matter. They will just polymerize. You know, they basically so it's growing, right? So you have the first layer of, so you have to do multiple cycles and get rid of any unused phenols. And the nice thing about phenols, talking from variables point of view, polyphenols is found in green tea and in coffee. So it's a natural compound as well. So if you want to make wearable devices, that's one of good candidates for making wearable devices.
Okay. So another another question is like the glucose sensors we usually use at home. Those like available in Amazon. Let's say is, what are those Aku something like Aku check, and another one is.
Are we talking about glucometers or are we talking about CGMs?
Yeah, glucometers. So
Yes.
they are all third generation, or
No, second generation.
they are all second generation.
Yes.
Okay.
Third generation is the Abbott ones, which are
Abbott
sitting there and measuring continuously.
Oh, okay, continuous. Okay, so but still like you are not puncturing anywhere, right?
It is subcutaneous.
Oh, okay. No, because I have seen some like ad and all, like you wear a watch kind of thing, and then is it puncturing really?
Watch is not, I mean, I don't think there's anything that's available behind a
I've
watch.
seen, like if you Google me, like you search some image and all, it comes like this watch kind of thing, and that also measures the glucose level, real-time glucose measurement.
Yes, but that's not very accurate, because again there's a problem of interference there. Optical detection of glucose and is much more harder, because you have to then be, you need a proper filter to be able to get that exact wavelength, otherwise you will have much more
so
interference.
That optical based probably is a first generation, measuring H2O2 level or something like that. Okay.
Yes.
No, H2O2 also.
And that makes the whole instrumentation bulkier.
So mainly he was interested to know whether there is any reaction like H2O2 or any sort of reaction in optical sensors.
I don't think so.
No, it's mostly spectroscopy based.
Optical does, so they use what they call a surface enhanced Raman spectroscopy. That's very sensitive. They use, but then nothing goes inside the body. It's all on the surface. They measure it in the sweat. They measure it through the surface. If you have surface enhanced surface I think it's called seesaw or something like that. So people use those kind of techniques. But nothing is as effective as the subcutaneously implanted sensors or the wearable devices.
Okay. Any more questions from the audience? Yeah, please.
Osmium tetroxide, yes. It should be second generation. You're right. It should be second generation. I mean, this was Abbott has got clearance in the last 10 years or so, and third generation the work has been going on for a long time. I mean, the rationale behind third generation was that we should get away from these mediators and work on something wherein we use engineered enzymes which can directly transfer electrons. So you're not talking about the existing enzymes. So if you remember, I gave you the example. You have to engineer enzymes so that the electron transfer units are not far away from the metal surface. If they're far away, because if you should look at the model of glucose oxidase, it's a big molecule, and the redox sites are far away from the metal surface. So when you immobilize them, there's a big gap there. You need to bring that to as close to the surface as possible, and these are the kind of studies that are being done in Professor Sode's lab. Chronoamperometric, yes. Yes. Yeah. So if you imagine you have pyramids, just imagine a pyramid. You have a very thin layer of polyphenol on top of the pyramid.
Okay.
Because when you talk about variables, let's say this is a wire. I mean, this is the kind of wire you would see in a subcutaneous one, the Abbott one. If this is a wire, this is coated all around. So sensing is a three-dimensional thing here. It's not a surface. It's not doing this. It's not in contact like this. It's around it. Does that make sense?
Yeah.
Does that answer your question?
Yeah, I got it. But still, like, I'm still wondering the question you asked everyone, like, this, what is the difference actually from third generation, like second to third? Of course, you remove the mediator part.
Yeah.
But then, like,
the third is basically where you have direct electron transfer from the enzyme
Yeah, exactly.
to the metal surface. You don't need a mediator, and
Yeah.
you're not relying on generating hydrogen peroxide.
How is it done? Like,
Yeah.
how is this direct
This is through engineered enzymes.
Okay, engineered.
That's where I gave the picture of Professor Koji Sode, who is engineering design. So these are not natural enzymes.
But anyhow, you need an electrode also.
Yes.
And
Yeah,
that
yeah,
should sense your, let's say, glucose oxidase.
Yeah. So you have enzymes which are naturally occurring enzymes. You do genetic mutations in those enzymes so that you reduce the size. It's like using fragments of antibodies. You don't use the whole fragment. Nanozymes. Nanozymes. Nanozymes is nanobodies and enzymes, but this is like using antibody fragments. So this is where you have something which has got the properties of an enzyme, the specificity, so that it will bind to glucose. It does the job of oxidizing the glucose, but when it comes to doing the electron transfer, it does not transfer it to the oxygen in the environment. It does not transfer it to the redox species that's there. It does the electron transfer directly to the surface. For that you need smaller biological macromolecules, so that the charge centers are very close to the surface, so that there is a much more effective charge transfer.
So that means you have to place where oxygen level will be very less.
Then it does not matter whether your oxygen levels are less or not, it does not matter then, because it is not relying on, it is a quick transport. It is like glucose comes in, oxidized, this one has got reduced, immediately electron transport. Sorry? Yeah. But the electron transport directly from the FAD to the, if they are closer to the surface, then rather than, if they are very close to the electron surface, then they will immediately transfer the electron. That is why they have to be engineered there.
Good evening, sir.
Yes.
Sir, what are FET devices?
What are?
FET devices.
Field effect transistors. Have you heard about transistors with a source and a drain? So these are basically, you have a source and a drain, and you're basically fixing the potential, and you're measuring or you're keeping the drain current constant.
Sir, is it same with the microneedle devices?
Same as the
Microneedling devices.
field effect. You can make microneedles also into field effect transistors. So instead of having working electrodes, counter electrodes, you can have a source and a drain.
Yeah, so what we do generally is we have something called as an extended gate FET, field effect transistors. So like you have microneedle, you can actually make the gate as the microneedle. So so there are 3 terminals in the FET, gate, source and drain. So the gate generally we make it long and use it as a microneedle. Okay, so in that way it becomes an active device. So microneedle itself is a, you can say, electronically passive device. But by using this FET, you can make the entire thing as an active device. So microneedle won't capture the signal unless you connect some electronics to it. The electronics becomes the microneedle sort of. We call it extended gate FET.
And I think there you're measuring the transconductance and things like that.
Yes, yeah.
Sir, actually I want to know that you already discussed about the third generation glucometer, and in which case we can use this type of devices, in which physiological conditions or in which type of disease, and when we can use these.
I think once you've, they are meant to be more efficient, but the most important thing is to have enough studies, because these are engineered with genetic mutations in the microbes to produce these kind of enzymes. You can use them in wearables as well.
Okay. So yeah.
Good evening, sir. So my question is, for this, like for IL6, you have designed MIPS by taking polyphenols. So, and if you take glucose, for example, so you have to decorate a receptor, like for enzymatic receptors or something. So if you are decorating the polyphenol here through electropolymerization on the microneedle, how are you designing, how are you decorating this sensor with the antibodies or the receptors?
These themselves are plastic antibodies.
So they have inbuilt antibodies in them.
I mean, we call them plastic antibodies because this is what they do. They are synthetic, you've basically templating a molecular a polymer to give you the shape and size of a bioreceptor there.
Yes,
which
so,
is
sir, that I know.
Yeah.
For example, MIPS will target the molecule that has the shape of it and all
Yes.
that. So in that case, we do not need an antibody or any other receptor.
No, we don't need
But
any other receptor.
if we are targeting through some other antibody, for like glucose, as you said.
Okay.
So then how will we design that?
In that case, you will take your monomer
Yes, sir.
and you will mix it with glucose instead of.
No, she wants to, I think, just to help you out.
Yes, sir.
She probably you want to coat the polymer
Yes,
with
sir.
antibodies. right?
Antibodies. Yes, sir.
So she wants some sort of an example where you have a polymer and an antibody on top of it coated, instead of using MIPS. So let's say there are some molecules which may not be customized for MIPS. Let's say maybe we could think of immobilizing antibodies on polymers. So if you have some examples on that, maybe you
Antibodies on polymers.
could polymer substrates or yeah for sensing
Just the microneedle.
on just microneedles. So
Well, people have done antibodies on microneedles.
so any example for that? Do you remember off the top of your head?
I think a lot of people rely on something called as thiol chemistry.
Okay.
So your antibodies have got a lot of cysteine groups, sulfur containing groups, and the nice thing with thiol chemistries is that you get self-assembled monolayers. So if you have, in that particular case, you have to make microneedles which have got a layer of gold, and then you can self-assemble the gold. The problem you will have is the following. You need to understand, you take your microneedle and you insert it into the skin. Can your functionalization protocol stand that robust process? Is it robust enough to stand that insertion process? Because when you penetrate the skin, there's every possibility that you may dislodge, because I've seen this in my own clinical studies. I used to have a nice polyurethane membrane. As soon as I would push it into the skin and take it out and then do a scanning electron microscopy, I could see that the whole membrane would be dislodged. That's why I'm trying to use polyphenol and polypyrrole, because these are thin films, but they're very stable, very conformal. So when you push them into the skin, they go in as in one unit.
Thank you, sir.
No worries.
Right. Any more questions? Yeah. So I have a few basic questions. So you mentioned about a thumb rule regarding the area of the electrode. So what is it? Can you please repeat it?
So your counter electrode should be twice the size, should be bigger than your working electrode.
Yeah.
And it ideally should be 2 to 3 times of your
Working electrode.
working electrode. That's why if you look at the designs, people have a dot and then they have a much wider.
Semicircle.
Yeah. This surface area is much more than your working electrode.
It's because to enable better current transmission.
Because you're at the end of the day, you're applying potential between the working electrode and the counter electrode, and your reference electrode is holding that potential.
Okay. So another question. So if you go back to the slides where you are showing the lactate and glucose results, yeah, these ones. So what is the cause? What do you think? Why it is saturating at towards the higher concentrations? Is
That's
it
the
lack
enzyme
of
property. So this is an example. I mean, I haven't gone into details for this one.
Yeah.
So what happens is that when you have your enzyme, in this particular case, if you look at this one here, the enzyme for the black curve where you don't have any membrane, the enzyme's Km value is quite low. You can enhance the Km value by having a membrane. What happens is when you have a membrane, you then you basically make it a mass limited. You limit the amount of lactate molecules that go through, and you slow down your response time increases, but you can cover a much wider range.
So it's like crowd control,
Yes,
yeah,
it's like a crowd control. It's like using a membrane there
Okay.
to kind of control the mass transport of your lactate molecules to the sensor surface.
Okay. Yeah, sir.
So probably that is why you told that you have to design your what is that sensor accordingly, so that it should
Meet
be
the
in
dynamic
between
range.
that dynamic range.
Absolutely.
Okay. So this is one of the way, like
This is one of the ways of doing that.
But is that the pores of the membranes are so small that it can really regulate the mass transport that easily?
if you look at, if you go down, if you kind of zoom in and go down deep down and start comparing the molecules and things like that, you need the membranes to have a size, pore size which only will allow diffusion of glucose molecules compared to albumin and those larger molecules. There's a lot going on there.
Thank you. Thank you.
Okay. Any more questions? Okay, I think it's a good time to wrap up this session. So thank you very much. So we can actually, so we will actually resume in 15 minutes. We can take a break and resume in 15 minutes. Yeah, thank you.